morning to one and all. I am welcoming you all for the Vishwadarshan lecture series, in which we are having very eminent personalities throughout the world. We are showing. Today also we are having a guest, a very eminent person who has worked in various fields, and even when he is having some physical discomforts, he is very eager to address the students, and the way he is expressing, I was very much impressed from the morning during my interaction. So I would like to welcome, on behalf of the University and the Department of Chemistry, I would like to welcome Dr. Ramesh Visvanathan. He is basically a PhD from IIT Delhi, and he did his postdoctoral degree on Crown Ether from the Texas Tech University. Presently, he is the director and head of QA Alliance Biosciences, Pondicherry, which is working very closely with the DuPont. So they are working on the probiotics. So probiotics now, I think all of you will be knowing the probiotics. Probiotics, what are probiotics? நம்ம எல்லாரும் தயிர் மோர்லாம் சாப்பிடுறோம்ல, all of us are taking curd and buttermilk. Why it is told very important? That is, it's very healthy. Why it is told? Because we are having many probiotics in that. So he is working on that. One of the leading companies in the world. He is working for that, and he has worked for Pfizer India. All the positions he has held as a director of Q&A. In analytical head, he has worked in Pfizer India, and now it is known as Zytus Pharmaceuticals, and he has worked for Syngene. In all the places, he has held a very high places, and he is known for its efficient work and ERP implementation, SAP planning, everything, how to use the resources in the company, effective use of resources in the company. So we are having a very eminent person to address us here. Welcome, sir. On the behalf of the university, I take the pride of welcoming you. Now I request Dr. Department to honor him with a stroll and a memento.
Password is 1961. This is just for information. I'm going to be talking extempore, so don't you worry. So, good morning, one and all. And I'm Dr. Ramesh Visvanathan. As he told, I'm 64. I had a fracture of the back, so somehow I managed to travel to about 2 hours. And coincidentally, I'm also a musician. So, me and my wife sang in front of all the 3 acharyas at variety of times. So my wife is coincidentally talking on music in your library building, and so essentially the reason I picked this drug design from mouse to man is I wanted to make the journey of what you do in the lab and how does it get translated into a hospital or things like that. How a man or a woman can get drugs. So essentially speaking, I picked this topic because it covers several areas like chemistry, biology, physics, chemical engineering, computer science, you name it, veterinarians, medical doctors, so it covers a toxicologist, so everything. So I just want to ask you one question. How many of you are chemists? Chemists, doing chemistry. How many of you are engineers? I don't differentiate between chemists and engineers because when I passed out of 11th standard, I got into IIT Kharagpur in ceramic engineering. My dad didn't want me to go to IIT Kharagpur. So, and coincidentally, when I also got into medical school in Bombay, medicine. But for me, cutting a frog was also difficult. Human being is too far-fetched for me. So I told my father that I don't want to do engineering, nor I wanted to do medicine. So I just wanted to do chemistry. I loved chemistry. Till date, I love chemistry. So many people said, you do chemistry, you can work in a bank, you don't have any bright future, nothing. So I disproved everybody. And today, I worked in the leading pharmaceutical company called Pfizer Corporation in the US, and which made the corona vaccine. And coincidentally, I was just telling Santanam sir, I was one of the experts in India to have released the corona vaccine from a testing point of view in the year 2021. At least I made a small contribution doing chemistry to the humanity. So essentially, why I want to tell you is I want all of you to be enthusiastic in the subject which you are. You like it. There are some people who have the talent to do engineering. Some people have the talent to do medicine. Some people have talent to do physics, chemistry. Strive in what you want to do. Do the best. And my time, there was no computer. Except books, there was nothing. You literally had to go to the library, spend Saturdays, Sundays, reading, writing notes, putting it to your memory, and that's how it is. Today, in a click of a button, from the cell phone, you can actually Google and figure out many things, right? So, technology has advanced today. So, why not utilize this technology, which is why I want to tell you that science and technology today are all going together. And also, my son, who is in the US, he did his, after 12th, he did his undergraduation. And why I'm telling this is, I'm just wanting you to be enthusiastic in what you want to do. So when he was in the 12th standard, he told me, Dad, you are working for the best company, you have passed out from IIT Delhi, you are working for Pfizer. Why you want me to study in India? You have money? Let me go abroad. So I had no answer to this question. So I sent him out. So 12 years, he's still there in America. He finished his undergraduation in biology and biological sciences. He was like me, didn't like engineering. He said no IIT, nothing. So no medicine. So he also loved biology. So he finished his bachelor's in biology. He did his PhD in Duke University, Durham, North Carolina, in brain cancer. So he finished his PhD, and today you will be surprised. He is at Harvard Medical School, working as a principal scientist, working for a 2019 Nobel laureate. So you can well imagine, and he has 4 papers in Nature. Just about 10 days back, he has published a paper on cell, and that cell paper is which is what we showed it to Vijayendra Saraswati, and he looked at it. He knows my background, and that's how I'm here. And his name, coincidentally, is Vijayendra, named after the third from. So we have a lot of what you call affection, gratitude for the acharyas. And when Vasanji asked me, I couldn't say no. I immediately said yes, will. Okay? So here I am going to talk. So I'll touch base wherever chemistry is there. I'll tell chemistry, biology, chemistry, chemical engineering, and things like that. So there may be some slides which I will skip. Don't bother about it. What I talk is more important. So if you want to write down, you write down. If you want, I'll share this presentation to your group. And maybe when I come next time here, I will definitely talk on probiotics, which is what I do today. So from a chemist, I become a microbiologist. At the age of 64, I know as much microbiology as a person who's worked in the microbiology field for the last 30 years. So you can imagine that the thirst for knowledge is still there in me. I'm enthusiastic. I love my subject. I love my job. I love what I do. And I want to do something good for this country. And hopefully my son is going to take us to the US. So I'm going to be settling in the US at some point of time. But yes, even if I settle, I'll be interacting with students and universities and things like that. Okay? So drug development, bench to bedside, mouse to man. So when you take a pill, I'm going to ask you a question. When you take a pill, what happens? Can any one of you tell me? When you take a pill, what happens? A tablet. What happens? This is a capsule. This is a tablet. So what happens when you take a tablet or a capsule? You have a headache. You want to get rid of the headache. What happens? It's going to be interactive. So apologize. I'll grill you. Don't worry. So what happens? Huh? Who told this?
ADMA processes.
Okay. Good. Anything else? So first, when you take inside, it disintegrates, dissolves, goes inside, and goes to the site of action to the bloodstream. Okay? That's called as bioavailability. So a drug is available to the body and goes to the site of action. When I say site of action, I have a headache. Just 10 minutes back, I took a headache tablet. My headache is okay. So it's called bioavailability. So you can imagine a drug is a chemical compound, goes into the bloodstream and is available to the human being. So the pharmacies, pharmacokinetics, etc. So when you take, drug gets absorbed, distributed, metabolized and excreted. If you take a drug and if it doesn't get excreted, you are in trouble. Okay? So introduction to drug development, basic research, idea generation. So when I say basic research, is there a Nobel Prize for engineering? Is there a Nobel Prize for engineering? Any one of you can tell me? Basic sciences, chemistry, physics, biology, come medicine. So there is nothing. So in America, if I tell somebody that I am a chemist, I am a scientist, they will be far away from me because they think scientists and chemists are high-tech individuals. If you say you are an engineer, they say you can come close to me. So for them, basic sciences have high demand in America. Why I'm telling America is my son is there, I was there in the US. So high demand, lot of money, and you require science, you require research in science, right? If you cannot do research in science, 10 years back, 10 years later, there is something which somebody is having a disease. If my son is working on kidney cancer, which is probably in a high rate in different parts of the world, if you can come up with a drug molecule to take care of kidney cancer, that's it. That's fundamental research. So fundamental research is very important. So the idea, what I'm trying to tell you is, each one of you are going to be placed at some point of time after you graduate, be it engineers or chemists or scientists, and at some point of time you're going to be interacting, being in an industry. So today, some of you can come to my company as trainees. I'm welcome. I can welcome you all and train you on how we do medicines. You're welcome. No issue. Only thing is, we'll have to fix up a time with your training department and development department. I can explain to you everything. So what I'm trying to say is very, very important that basic science, basic research, idea generation is important. And when you develop a drug, how drugs are discovered and developed. There's one guy coming out of the haystack saying that I've developed. You know how long it takes to develop a drug? 8 to 15 years. Okay? When I say drug, a cancer drug, which is going to be taking care of colon cancer or brain cancer or kidney cancer, whatever it is, it takes 8 to 15 years to come up with one molecule. One molecule. And sadly speaking, I should tell this, but it's important I tell this to you all. We say we are some of the best scientists in the world. We are the best scientists in the world. I disagree completely. 74 years, we don't have a single molecule which we can say made in India. Whatever you see in the drugstore is nothing but a generic equivalent, a copycat of a drug made across the world. You understand? So they spend millions and trillions of dollars in coming up with one molecule. So to come up with one molecule, what is it takes? How many people are involved? How many areas are involved is what I'm going to talk today. Am I clear? Correct? See. This is the bulb. This is the discovery phase. Then you have a lead or optimization phase. You have to do preclinical trials. So preclinical trials is basically a clinician, a doctor, a volunteer. You have to do the studies in a hospital where human healthy volunteers enroll themselves, and the drug which you are going to be developing is given to these human healthy volunteers. And they take it, and the blood goes in the bloodstream. You take the blood out, the plasma which is there, you check for the absorption distribution, ADME as madam said. So you check and see the bioavailability of the molecule. So it's very easy to, for all of you. I was one of the scientists to have developed metformin. Maybe I was there for 3 years, but drug took 10 years in the globe to come out. So what you see in the chemist shop today is not made in India. It's only a copycat, generic equivalent of what is made across the world. To develop, you require money. So, money is everything. In the sense, you can go to a Josian, you can go to Sai Baba, I see serial Sai Baba, he puts that ash in the water and gives you become alright. Those are all cereals. But this is reality. Okay? You understand? Bench to bedside. So, first you have obstacles. What is the obstacle? Some disease. Then you do scientific study. You have a regulatory. A regulatory in the sense, some drug agency which actually wants to regulate how you have made the product. Then ethical. You do need to do things in an ethical way. You can't do in a cacophonical way, then finance is important. How much money is involved? So I'll just take you back to 1961. Why I want to take you back to 1961? So, pardon, these slides are just for information which I can give it to you. What I talk is very important. In 1961, there was a drug called thalidomide. Okay? Thalidomide was given to pregnant women to take care of the morning sickness. Okay? Women used to vomit and all that during pregnancy. 1961. There was no research done that time, in 1961, on clinical studies, nothing. That drug was made, given to women. The children born out of the mothers died. Close to 4,000 children. This is in the US and UK. Then John F. Kennedy, the President of USA. Why I'm telling you all this is very important for you to understand the science behind these kind of things which have come up today. Then the President John F. Kennedy called all his ministers and said, stop it. Come up with a solution. And I don't want this cacophony, unethical stuff. And coincidentally, the drug thalidomide, its isomer, the drug and its isomer. The isomer was an impurity. The impurity is the one which killed the child. So, and the lady who came up with this clinical studies and this 8 to 15 years business, this lady at the age of 94 was honored in the US for her contribution to coming up with this road from mouse to man. You understand? So it's not easy. So she came up with a logic as to how to do it. So from a quick fix solution in 1961, today you have this 8 to 15 years. And corona was one of the kind where you can't wait 15 years, right? People are dying, right? So vaccine, they came up with a fast track method to develop the vaccine in 2 years. Pfizer, where I work, came up with a vaccine. AstraZeneca in UK came up with a vaccine. And today, in India, we only re-engineer. The development was done in the US. In India, Bharat Biotech and Serum Institute in Pune only manufactured the vaccine. Modi may say we have saved the world. No. That is Modi's understanding. My understanding is those people who made the drug have saved the world. We only re-engineered and gave the product in India. And in India, I think some of you must have taken vaccines as a child, right? We have all taken BCG vaccine. I think all of us have taken BCG vaccine. BCG vaccine was the one which actually gave high immunity to the Indians. So that is one of the reasons India had low death rate during COVID than anything. But unfortunately in India, the government, the politicians give wrong messages. As a scientist, I don't believe in politicians. I believe in science. You understand? So, doctors fleeced the people. Everybody was completely thrown out and number of deaths were less. Number of deaths in America was more because they did not take BCG vaccine. You understand? See? So you look at 10,000 compounds. For one compound to come up, you have to look at 10,000 compounds. And how do you look at 10,000 compounds? You synthesize these compounds in the laboratory using a drug design module and also do computer molecular modeling. This is where the technologists and the scientists come in. So in the early stage, if you want to come up with one molecule, you've got 10,000 compounds. So these 10,000 compounds, they study the biological activity, they synthesize the compounds in milligram level. So from 10,000 compounds, it comes to 250 compounds. Then out of 250 compounds, 5 to 10 compounds shows promise, and after 5 to 10 compounds, 2 compounds can show future promise, and then 1 compound goes through clinical studies, goes through FDA, that is Food Drug Administration, which is a regulatory agency, and the FDA approves in the US. So this is the life cycle of drug development and its timeline. Okay? So you can well imagine, and you can make a compound, right? But if you have to make a product, you have to make what you make is a chemical compound, right? The chemical compound has to be synthesized in the laboratory, tested in the laboratory. A person like me tests the laboratory, this thing, using IR, NMR, and other things, which the chemists, all of you know, to be tested. But it has to be made at a large amount in tons. So who will do it? Engineers. This is where the engineers come in. Chemical engineers. Okay? Fermentation, biotechnology, fermentation group. So I'm just trying to tell you the departments which are involved in the whole exercise. So today you cannot say I'm superior, you're inferior. No. Everybody is important in this. I picked this topic because I just wanted you to understand the sequence, the areas which are involved in the drug design and development, and why it is important. Okay? The process. Again, 10,000, 250, 5, and that is the bottle. Bottle, or the, what do you call, the strip I showed you. Okay? So it does the whole exercise. And everywhere the FDA is involved in this whole exercise. Okay? Where does it start? Discovery, lead optimization, preclinical, phase 1, phase 2, phase 3. I'll tell you what are these phase 1, phase 2, phase 3. Why this 18 years is how I have split up this 18 years. And lastly is the clinical testing, followed by commercialization. So discovery, idea generation. So you can use AI, but brain is important. AI is a tool to speed up, but fundamental brain is important. That's where you have to learn science. That's where basic science is important. One good thing in India compared to the US is the fundamental science is extremely good in India, which is what I like. My professor used to tell me, you don't talk, you tell a story. This is what my US professor used to tell me when I was in the US. So I'm not talking, I'm telling a story. You understand? So, fundamental education is excellent, but advanced research is superior in the Western world. You understand? Why? Because they have money. So, chemists, biologists, physicians, and you usually, a chemical is synthesized or naturally occurring. You identify a problem that has to be fixed. Is it cancer or whatever it is? You do, and then novel compounds, recycling of old drugs. There are certain compounds which have been developed, but may not have been studied for a certain thing. So you can reuse the compounds which somebody has made. So you look at new compounds, old compounds, then natural compounds, and in drug development, most roads lead back to chemistry. You understand? So, chemistry, biology, science is very, 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 very important. This is what I'm repeating again and again. Don't even ever think that I'm a chemist, I'm going to be a banker. No way. If you have the will, you will hit the right block at some point of time. Okay? Target validation is a critical step in drug development. You can see how first-in-class drugs according to molecule type and target family, discovery targets. You look at the discovery targets. and discovery high throughput screen critical step in the process, results in starting point for a drug design, small molecule or a biologic, protein, DNA, sRNA, screen libraries with millions of entities. There are libraries available. You can pick molecules from the library, which is where the artificial intelligence helps you. You can ask them to pull out the compounds then. Pull out the compounds. So based on the compounds which is pulled out, you can, otherwise you look at million compounds, right? Who has the time and energy to look at million compounds? Save time using artificial intelligence. Come up with 10,000 molecules. 10,000 molecules, do small synthesis, come up with 10 molecules. So screen libraries, robotic and automated, start the chemistry process, results in a compound that chemists can modify. So you have to modify some of the compounds which have been used. Sometimes, they made it, but then you want to mimic some other compounds, you can actually modify a compound to come up with a better thing, which what was made 20 years back, may be applicable now, which is modified. See, library of compounds. I'm just showing a rack to make you understand that huge depository of information is there, chemical information is there, which you can actually pick up, just like that. Chemical structure, purity, quantity, physico-chemical characteristics of compound, where the IR, spectroscopy, NMR and all will come into virtual computer-aided drug design. Most important. It saves a lot of time. Okay? Today, with AI, my son is using AI. He is working with Harvard Med School and also with MIT, Massachusetts Technology on bioinformatics. So thanks to him, his Nobel Prize professor is taking his knowledge and learning which is learning in MIT and Harvard to use technology to an advantage. You can understand. But nobody taught him, he learned. Correct? So nobody will teach you how to learn. And if you have to learn, people like me in the industry is willing to help you in nurture your understanding of chemistry as to what we do in the plant. You are welcome. See again, CAT, 10 million, biochemical, cellular, 1,000 compounds, specificity, in vitro screening, preclinical testing, so on and so forth. Which will take how many years I split up. So a target compound, 2 to 3 years, lead improvement, it takes 3 years. Then mechanism of action, how the drug goes into the human body. Mechanism of action, that's important. So drug and sometimes the metabolite can be useful. A compound where I work, Pfizer, is called atorvastatin. Atorvastatin is a cholesterol lowering drug. Okay, I think some of you may have taken statins. A person like me with an age, statins are easily there. So atorvastatin, you will be surprised, is a compound made by Pfizer. You know the amount of sales which that compound generates in a year? 15 billion US dollars, one compound, which is more than the size of all the Indian pharmaceutical industry. It took them 10 years to come up with atorvastatin. So today it may take 10 years. Once you hit it, you become a millionaire. That's what it is. You understand? So fundamental science is important. Basic science is important. So we come to lead optimization. So when I say phase zero is 10 to 15 subjects. Subjects is nothing but patients, human healthy volunteers. Okay? First you do it with human healthy volunteers. Then you do with phase 1 is 20 to 50 normal volunteers again. So we put them in a clinical laboratory There are doctors monitoring whether they took the drug or not. They keep taking the blood samples, analyze using HPLC mass spectrometry to understand whether the drug has actually gone into the human body and whether absorption has taken place, metabolized, excreted. So all this is understood. Bioavailability is done. So it is conducted by a clinical pharmacologist. So chemist, physicist, biologist, medical field, veterinarians, pharmacokinetics and pharmacology. See the amount of one molecule can rip up the entire world. How many people how many branches are involved? Then phase 2 is 100 to 300 patients. Then phase 3 is 250 to 1000 selected patients to establish. Selected patients means sometimes person having a disease, whether it is actually working or not. Okay? In human healthy volunteers, you understand whether the drug has reached the human body. Okay? But there, you understand whether the disease is working. Okay? And there, it is ethical. You need to get signed from all the people who have volunteered. Tomorrow they take the drug and something happens to them. They'll sue you, right? Yes. You gave me, but one person died. We are done. Even one person dies, the drug doesn't move forward. So millions and trillions, one compound you make, somebody dies, stops. There ends the matter. 20 million, 500 million, down the drain. Okay? So it's not easy. It's risky, but enjoyable. Phase 4, post-marketing surveillance, 2,000 to 10,000 patients, which is where the drug is made and given to the patients across the world and taken a survey. Post-marketing surveillance, the doctors are involved, the chemist, the pharmacist, the medical representatives who work in the drug companies go and take a post-marketing surveillance. Whether the drug is working, is somebody having an adverse effect, things like that. But more often than not, you take a drug. Unfortunately, this is important that you should know. When I take a drug, right? I'm a diabetic. Very many times when you take a drug, because you take multiple drugs, the drug which I may take may not be effective to me because I am a diabetic. You understand? So, in the world, no one drug can cure all the millions in the human world. You understand? Because each person's metabolism is different. You understand? Sometimes you take, a Vala Pandla, because, so this, the doctor should know. And who will educate the doctors? People like me and others who are doing drug development. We understand the mechanism. We study the patients, understand who is taking what drug, and then come and analyze, saying that, okay, if you see in this drug is not to be used for lactating women. Pregnant ladies should not be taken. These are all information which is put on that bottle. So lead compounds, chemical synthesis, drug in use, new compounds from plants. You can actually derive a lot of things from plants. Molecular modeling, silicon graphics. So I worked on silicon graphics in the US. So basically you can actually, the molecule which you are targeting, you can actually draw the structure on the computer. So we have all tools now to draw the structure on a computer and just say, model it. So this is actually a molecule which actually it looks like, how the molecule looks like. Whereas what you see maybe something on the right. Okay? This is actually what it is on the left side. Actual molecule. This is what is called as solid state molecule. Also called as Corone, Pauling, Linus model. Corone, Pauling and Linus model. And Linus Pauling is the only person in the world, apart from the Curies, to have got Nobel Prize twice. He got for chemistry, Pauling exclusion principle. Any of you have read in chemistry, Pauling exclusion principle? And he got another Nobel Prize for peace. CPK model, Korun, Pauling, Linus model. CPK model. So I have this actual model in my house. So to understand, I can play around. I have a computer also. I can design this in the computer and see how it looks like. Whether the oxygen is in the front or back or whatever it is, you can understand. Must know final goal of the drug for its step. To define parameters, target disease, acute and chronic administration, best route of delivery. Is it intravenous? Is it subcutaneous? Is it intramuscular or is it oral? And in oral, do you want a capsule? Do you want a tablet? Do you want a dry syrup? Whatever it is, you have to design. So this is where the pharmacy group and the chemistry group will come in. And see, person is taking, and he said, somebody said it disintegrates, dissolves, goes into the human body. This is the lead optimization one. Assay is used during, assay means whether the compound is really 100%, so you have to check by using analytical tools like NMR, HPLC, etc. And then pharmacokinetics, where plasma half-life with specific route of administration, brain penetration, and tissue distribution. Is it distributed in the tissues? So this exercise is done by a veterinary doctor. And basically, where does the mouse come? You can't give the, when I say phase 1, phase 2, etc., directly you cannot give to human beings. Correct? This is where the mouse comes. So early on, my son every day has dedicated 200 mices with him. He goes and gives the drug, and he cuts. For me, even to hear this is because I can't cut a frog. He tells me every day he cuts a mice, takes the liver and other things which is close to a human, checks, and once the mouse model is okay, and in the mouse model we look at toxicity, whether the drug is toxic, whether the drug is cancerous, most important, because it's a chemical compound, right? Is benzene a cancerous or a non-cancerous substance? Benzene, you know, no? Right? Is it cancerous or non-cancerous? That's right. So you can well imagine, 24 years back, I was using merrily benzene in the lab, but I didn't know that it was a carcinogen then. Today, benzene is replaced by toluene. So even though all the drug molecules, the basic structure will be benzene, but built up. When it is built up, it is not a carcinogen. Benzene per se is carcinogen. So you cannot use benzene as a solvent towards manufacturing of a drug. You have to use toluene or other mediums. You understand? So you do in vitro screening, in vivo screening, both actually out of the, on the bench you do, and on the patients you do both in vitro, in vivo. And in vitro, in vivo is actually correlated beautifully in the pharmaceutical industry. Today, whatever drugs you get today in the shop, most of the companies, sorry to tell this, but this is the reality. When you go to a pharmaceutical compound to buy, pharmaceutical drugstore to buy a drug for you, no bioavailability is done in India. Nothing is done. It's just randomly made, given to you, and you also take it. But in US, if you have to make a drug, it has to be bioequivalent. So I make a compound. I've spent millions and trillions of dollars in making a compound. A similar looking compound you make, you have to make it bioavailable. In the sense, it's the same compound. I make a compound similar looking, it has to be bioavailable. So when it is bioavailable, it's only thing, it is safe pure and effective. But nobody in India, domestic market, does any bio availability or bio equivalence. Saturated oils. But Bhagavan Pathakaran, number use pannom. I am only telling you the facts which are happening in today's world. Don't get scared. But இது பண்ணணும் பண்றது இல்ல. See, this is the candidate summary so far. So mouse model, compound screening, target validation, mouse this thing, compound screening, secondary assay, in vivo analysis, again mouse is a this thing, and it goes to clinical studies, preclinical development, and their clinicians, veterinarians, all these people are involved. Toxicology, maximum tolerated dose. In a compound, in a drug industry, can you take paracetamol a day சாப்பிட முடியுமா? சலவலின்னு சொல்லிட்டு 5 டேப்லெட் சாப்பிட முடியுமா? முடியாது. ஏன் சாப்பிட முடியாது? Maximum available dose. Whereas in a probiotic is only in the gut, doesn't go through the bloodstream. It only kills the bad bacteria. Entire gut has all bad bacteria. So in our company we make live good bacteria. So when we take the live good bacteria, it replaces the bad bacteria in the gut, makes you healthy. And if you are healthy, half your problem is solved, right? When you go to eat food outside, you are fussy. தண்ணி எப்படி இருக்கும், சாப்பாடு சரியா பண்ணிருக்கானா, எண்ணெய் சரியா பண்ணிருக்கானா. These are all things which you look at, no? Road side சாப்பிடுறதுக்கே நமக்கு கஷ்டமா இருக்கும். You think 10 times. And சாப்பிட்டுட்டு எதனா ஆச்சுனாக்கா, oh, I had this food outside, this may be the problem. So probiotics ensures that this is all taken care. So that's the other side of the story, which I am working now. So ADME, absorption, distribution, metabolism, excretion. This is all done through HPLC, mass spectrometry, and clinicians. They have to check whether the patient BP is okay, heart rate is okay, blood samples seriya irukanu papa, and patients, one set of patients is given a person who's made the drug first time. The other set of patient is given by the Indian company. Then after 15 days it is swapped. The person who has taken the foreign drug is given an Indian drug. So both the patients are analyzed. Both the set of patients are analyzed, and then you say it is bioequivalent. It costs how much, you know? 1 crore of rupees to do this exercise. How many companies are willing to spend 1 crore of rupees? Unfortunately. I'm part of the industry. And about I've also gone through this. Tolerability usually start with rodents, mice and rats. You don't use primates. Monkey la use pandradhu ipo. It's banned. Monkeys are banned across the world. Only in few cases in US, monkey is used. My son has gone to medicine and worked with some monkeys for his kidney research. So only, but that too again, the committee has to approve that he can do this research. Adukapram da paada mudiyum. Outbred inexpensive mice. Rule of the thumb in cancer: 20% of the body weight loss. LD10 is MD6. So rule of the thumb: if the weight loss is 20%. You can't use the cup. So they weigh the mice. Tolerability. So all these things are checked. WBC, RBC, platelets, amylase, lipase, glucose. The patient who does the clinical, all these tests are done. Like now blood test பண்றோம் இல்லையா? இதெல்லாம் செக் பண்ணுவாங்க. So it is so scientific. So scientific. Now, madam, you can explain? Please, let me take a break. Beautiful. I love this slide.
Me too, sir. Sir, already our third year students having this paper, sir, pharmaceutical chemistry paper.
Okay.
They have this unit, sir. Have this in our unit. See, good afternoon to all. Already we know our third year students already aware of this ADME process. ADME process is nothing but once we are taking a tablet, after taking the tablet, we thought it is one of our students said it is dissolved. Yes, obviously it will be dissolved. That means the absorption process takes place. First one, absorption A, then second one, distribution. Then it will be distributed wherever it want to go. See the picture. See, once we are taking that through the mouth, we are taking the tablet. After taking the tablet, it is coming, see, through that blood vessels, it transforming, it distributed that through our blood vessels, it is distributed the drug. After that, the third one, metabolism. Already sir said, through based on our individual metabolism, it will be that the processes, that how it is, the action will be taken through the metabolism. At last, elimination. See, all that we are taken, excretion process is completely taken out. We are the healthy person. If it is not, we are the patient.
Correct. And most of sometimes what happens when you take a drug, no?
Thank you, sir. Thank you for this opportunity.
When you take a drug, sometimes or 2 hours the urine becomes yellow. Don't worry, nothing has happened. It's excreted.
Excretion process taking urine,
Through urine
sweating.
only. If it does not excreted, you are in serious trouble. There's a high chance of you passing away. So 80% of the drug passes through urine. I'm not scaring you, I'm telling you the process. See, this is a lovely slide. I love this one. Optimization, poor systemic exposure, poor bioavailability. Bioavailability means it's not there. So then it goes to blood-brain barrier, plasma binding, metabolic, physicochemical properties. pKa, I was talking to sir on pKa, which I have developed a McFIP method. Solubility of the drug is important. So all these parameters will be studied. So it is science, pure science. And from pipeline until commercialization launch, for each actually marketed drug called as new chemical entity. New chemical entity. Again, remember, it's new chemical. New chemical means nobody has done it before. It's called NCE. There are been more than 1,000 substances that underwent screening in India. Without the use of available computer-based ADME filters, this number would be even larger. So you can see 10,000, 1,000 more compounds, 0, 3.6, 1.2, launch of NCE up to 10 years. So it's got a funnel. The efficacy models, relevant models, minimally effective dose, therapeutic window depends on the disease. And even in India, if you have to do what you call mouse model and all that, unless you get it approved by a clinical group, unless clinical group it's a combination of scientists and common people. Means you also, you may not be knowing science, but you will be also a part of the whole group. Lawyers, engineers, all of them will be part of that group. Two species and non-rodent, the monkeys. Biologicals, most relevant species, clinical groups and schedule. Pharmacokinetics is most important, optimal. So now is the final product. Okay? Capsules, your vials, your injections, things like that. Why do you take an injection? Why do you take an injection? Sometimes injection is by Why do you take an injection? Because, tell me, you have any idea? Madam, you can tell why injection is taken?
It's an immediate drug action.
இது உள்ள போய் டிசால்வ் ஆகி கீழே போய் பிளட் ஸ்ட்ரீமுக்கு போய் டைம் ஆகும் இல்லையா? ஸோ இப்போ 10 days back my wife had a diarrhea and things like that. She had what you call oral rehydration. So strength illa. So they have to give this.
Intravenous.
Okay? Quick cut. And இங்கோ அடிபட்டிருக்கு. நான் பண்ண டேப்லெட் கொடுத்து பாருங்க. Injection. So all these things are filed in a what is called as an investigative new drug application. It's applied in the FDA. FDA will review it. நாங்க டாக்குமெண்ட் எல்லாம் கொடுத்துட்டு டாக்ஸிகாலஜி ஸ்டடிஸ் கார்சினோஜெனிக் ஸ்டடிஸ் எல்லாம் கொடுத்துட்டு they will review it thoroughly and then only they will approve it. So in the 8 to 10 years bucket, of course ஒரு மாலிக்யூலுக்கு 8 வருஷம் ஆச்சுனாக்கா அது ஒரு கம்பெனி பண்றது. பட் ஆனா ஒரு கம்பெனி மேபி லுக்கிங் அட் 5 molecules for 5 different diseases. இந்த மாதிரி எவ்வளவு கம்பெனிஸ் இருக்கு. ஸோ அதனால இப்போ ரிசர்ச் இஸ் கோயிங் ஆன் இன் அ ஃபாஸ்ட் ஃபேஸ் வேர் அண்ட் all of them will rely on basic research, like what my son is doing. So my son's professor is a what I call a leading director in several pharmaceutical companies in Boston. So they take his help. And my son is going to what I call Antarctica and for understanding why fish is surviving in Antarctica with low oxygen. So a project கொடுத்திருக்கா government of USA. So he's doing the exercise. Clinical testing, commercial phase. See how many populations. These are all important. I give it. Of course, only it is that you can take it, sir. So first stage is testing in human subjects. Second, first use in combo in humans, human healthy volunteer, in cancer specifically, late stage disease, and dose limiting toxicity. You have to understand, can I give 2 doses, 3 doses? These are all studied properly. And when you take a tablet, sometimes the doctor says, take it before food, take it after food. So follow that instructions properly. Now, follow padlenna work agada. Then we complain. So different probability of phase 3 success: hematology, infection disease, metabolic, urology, allergy, respiratory, other endocrines. So these are all available information which is there in the library. You can actually look at and figure out how long your drug will take to be developed. So a few success stories which is there, how long. So discovery phase, clinical development, commercialization. So you may develop a drug. You may utilize it for something else. There's a drug called sildenafil citrate. Sildenafil citrate is used by men using for, of course, there's probably not an audience to tell this, but it's used in sexual intercourse and things like that. Okay? It was actually used for heart. The drug was developed for heart, but landed up as a sex. You understand? So, repurposing. It never went for heart. So, translation research, there are a lot of people who do reduce cancer incidence, morbidity, mortality, translation of scientific ideas into clinical practice. In clinical practice, use of scientific basic principles to push forward ideas. Definition of translation. This is lab, clinical, population, new tools, new application, and can be used in clinical and preclinical. So, significance of translation research is drug, pancreas, colon, pancreas, renal, prostate, cervix, colon, cancer, all kinds of, then some things are malignant, non-malignant. Summary, drug development exactly 1 hour. Drug development is complex and involved new targets, new molecules, better understanding of disease. Life scientists are the gatekeepers for interesting opportunities for drug discovery and development. When I say life scientists, people like you can think, are the important people for drug discovery and development. A laboratory observation is conceivably a starting point for a team of investigators with scientific, clinical, ethical, and societal implications. Bench to bedside. Clinical optimization you can do. You can do a lot of, as I indicated, you can do a lot of imaging, things like that. So I come to the last slide. Thank you for your attention. I think I must have spoken randomly from out of my heart, which is what I do. What is there is just for information. Any questions for me? Feel free.
Sir, please share chemist's role in the metformin drug development process.
Okay. Chemist role.
Yes, as your role. Your role.
So when the drug is towards commercialization, right? Towards
Yes.
essentially what you do, metformin is an active compound, but you have to mix the metformin with inactive compounds. எல்லாருக்கும் metformin கொடுத்தா, metformin is 500 mg in solra, but that 500 mg is mixed with starch and other things which doesn't affect human body. Okay? And metformin, most of the drugs are chemicals. But if you see the pack, it says sodium salt, metformin hydrochloride. Why hydrochloride? Because it's soluble in water. Because நம்ம தண்ணி எடுத்து தான் சாப்பிடுறோம். நமக்கு chemical compound எடுத்து alcohol கொடுக்க முடியாது இல்லையா? So when we make metformin, basically this is a good question you asked me. You have to understand drug has to come to the human body as quickly as possible. நம்ம பாட்டுக்கு ரிசர்ச் பண்ணிட்டே இருக்கலாம். No into it, right? Fancy பண்ணிட்டு இருக்கலாம். So அதனால we understand whether metformin has to be given in a patch, whether it has to be in a drug syrup, dry syrup, is it capsule, is it tablets, is it injection? என்ன மாதிரி dosage form first you have to understand. Then you develop the molecule with different types of excipients. ஒரு எக்ஸிபியன்ட் பண்ணி அது அவைலபிள் பிளட் பயோஅவைலபிலிட்டி இல்லைன்னா என்ன பண்ண முடியும்? யூ லூஸ் இட் 1 year. ஸோ ஒரு மெட்ஃபார்மின் பண்ணணும்னாக்கா ஃபார்முலேஷன் டெவலப்மெண்ட் பேசிக்கலி இன் தி pharmaceutical industry take up 10 different compounds. And another thing, the product has to be stable, no? நான் மெட்ஃபார்மின் பண்ணலாம், பட் 6 மாசத்துல புட்டுன்னு போயிடுச்சுன்னா ட்ரக், கரெக்ட்? So you have to do what is called as stability studies. The drug has to be stable to the intended shelf life. So if you see the label, it says 24 months shelf life. Manufacturing date, expiry date, right? So unless the metformin is stable for the 24 months, no point in developing the molecule. So you pull out stability. So you have to wait. So ஒரு drug பண்ணனும்னா 24 months wait பண்ணனும். Actually test பண்ணி எடுக்கணும் every month. So ஒரு product எடுத்து போச்சுனாக்கா 10 product பண்ணி do stability parallelly. Adala whatever is behaving best, you do the bioavailability. Okay? That bioavailability, of course, if you are an innovator, it's a bioavailability. If it is a lookalike drug, it will be bioequivalence. You understand? And adala, you will do the drug testing. Metformin will have impurity, like thalidomide. So it's very important that you understand what are the impurities in metformin, and that impurities have been controlled. How do you control? You take metformin, put it in acid buffer, basic buffer, peroxide, photostability, all these things. And then are the human body acid regulate and photostability. Some drugs expose panna poidum. So you do all these studies to understand how the drug is degrading. So in the pharmaceutical formulation, this thing, you do metformin analysis and impurity analysis and find out how many types of impurities are there, what is the level of impurities, how do you control them, how do you test them. These are all part and parcel of drug development. Drug development at that last stage, and the 2 varsham is a big. That is done by scientists, analytical scientists, formulation experts, pharmaceutical people, and things like that. Pharmacokinetics is basically pharmacology people. They are people who take the blood sample and do the bioavailability.
Thank you, sir. Sir, another question.
Sorry.
Sir, are you facing any biosimilar drug?
Well, many biosimilar drugs are available in India. In fact, to be honest with you, in the probiotic field, we have come up with a probiotic called pro-biosimilar. See the terminology. We are in the probiotic field. There is a probiotic which is developed by somebody. Okay, so it is biosimilar, but it's probiotic. So we coined the term pro-biosimilar.
But everybody said it is not safe to take.
See, it's all, don't go by, first of all, Google and other information is good for information. My wife has this habit. Now I was a scientist. And in the drug எடுத்துக்காதமா இந்த பிராண்ட் நேம் எடுத்துக்காத அப்படின்னு சொல்லுவேன். இல்ல டாக்டர் சொல்லிட்டு அது இதே தான் எடுத்துப்பேன். நான் சொல்லுவேன் வேற எதனா பிராண்ட் நேம் இருக்கும் அதை கொடு. I understand. See, being in the pharmaceutical industry, I understand the drug inside out. Which drug is stable, which drug is not stable, which company is good, which company is not good. But adhan. So there are certain things, women especially, it's very difficult to convince them.
This biosimilar also, it will take this that all the tests that the clinical.
Correct. But there are certain things which at the early discovery and the phase la poidu.
That's why I'm asking.
Adu poidu.
Then how it is safe, sir?
No, but you do bioavailability and this toxicity la podu. And the discovery then and the lead compounds la varade. And the 5 years.
Half of the work vandu idla skip agudhu.
5 years you will say. 5-6 years you will say.
Thank you, sir.
Any other questions?
Sir, I am PhD research scholar, sir.
Huh?
PhD research scholar, sir.
Okay.
So currently we are working on developing cyclodextrin grafted magnetite nanocarriers.
You are working with sir?
Yes, sir.
Okay.
So usually anti-cancer drugs giving many side effects, sir. So these side effects reduced by adding some additives.
Okay.
So we are developing one additive that name is cyclodextrin grafted magnetite.
Okay.
So we are in 4th stage, sir. Next stage is in vitro studies, sir. Once we completing this all laboratory procedure, we go for in vitro studies. So could you please suggest me any collaboration for doing in vitro studies?
I have to first understand what you're doing. If you can, if it's not confidential, you can share that information. I can, sir, you can share this on my mail, on the Gmail. I'll review it
Okay,
and come
sir.
back to you. And I'm not going to steal your product because I'm 64 now. Now, if I want to your product,
Okay, sir.
okay? So I'll definitely help you.
Okay, thank you, sir.
Any other questions?
Sir, can I know your thoughts and ideas about Ayurveda?
I don't want to comment on Ayurveda, honestly speaking, because of course you do have an Ayurvedic college and doctors and things like that. I'm more of a, my wife actually, sometimes somebody says, the Ayurveda is go seri I don't know. For me, the pharmaceutical medicines are science. And the science is theriyadu. So unfortunately, what I don't know, I don't want to comment on it.
Sir, I have another doubt. Thank you for the wonderful interaction, sir. So as you said, a drug takes 8 to 15 years to come to market, sir. But how come Covishield, vaccines like Covishield and Covaxin come to market so early, within 2 years?
It's a vaccine, right?
Yes, sir.
So it was released under an emergency. Millions of deaths were happening, man. Right? Can you wait for 15 years for a vaccine to come up? No, not. And another thing, the beauty is the vaccine is to prevent your susceptibility to corona.
Yes.
But during corona, what did the doctors give? If you had fever and all that, enna kudutha doctor? Anybody can tell me? Correct. Paracetamol kuduthe paisa padita. You understand? So this is what I'm saying. I am in this field. I actually love இப்படி ஒரு மக்களை லூட் பண்றாங்க டாக்டர்ஸ் ஹாஸ்பிட்டல்ஸ் லட்சமா லூட் பண்ணிருக்காங்க unwanted. Zinc. Why zinc? Because zinc gives immunity. Probiotics which we make gives immunity. So during corona time, our company was doing very good because immunity was there. You understand? So this was given on a fast track on an emergency thing. And it luckily helped. But my son, who is in the US, is coming up with a vaccine for one vaccine to take care of everything. You get it? This is what I'm saying. Research is, I thank myself and thank my son for what he is today, because at least at some point of time he's going to come up with vaccine. He's one of the lead, because having worked in Duke and other things, he's a vaccine expert also, a virus expert. So on the virus expert, he's coming up with a group of people to come up with one single vaccine to take care of all the viruses. Okay? So this is research for you. This is where fundamental science and basic science and basic research are important. Any other questions? You're good to go? So thank you very much.
What differences between the clinical trials for vaccines and other drug molecules? Protocol difference.
Protocol. That is basically, it was a 6-month study. Lancet journal, and the Lancet they publish all the clinical trials for COVID vaccine. So if you actually Google and figure out, you can get the result study. But நிறைய பேர் சொல்றா Pfizer fudged the data, fraud பண்ணிருக்கா, இது பண்ணிருக்கான்னு சொல்றாங்க. These are all people who have no knowledge of science who come up with this thing. Big company like Pfizer will fraud a data? No. Serious business. Serious. So now this 6 months study report. In fact, you would have come in the paper. It's undergoing clinical trials, studies, etc. So they have a protocol, separate protocol for vaccines. And they will do it first they will do it with human healthy volunteers, then with the corona patients. You understand? And in the corona patients, there were some reports saying that 2-3 corona patients died and things like that. But what ultimately, largely the vaccine helped the population safe, correct? 2-3 deaths ஆயிருக்கலாம், but ultimately at the end of the day, the world was suffering with corona, right? So it kind of helped. That's what it is. So sometimes you have to take a risk. Wait for 15 years or do a fast track. And only corona. Only the vaccine got a special treatment from FDA. FDA means US FDA, not everybody. You understand? Yeah.
நீங்க பிரசன்ட் பண்றதுல தான் ரிப்பீட்டடா வந்து இந்த ஹெல்தி வாலண்டியர்ஸ் வேர்டு வந்து யூஸ் பண்ணிட்டு இருக்கு. இது வரைக்கும் எங்களுக்கு அந்த கிளாரிட்டியே கிடையாது. They use similar talks basically clinical trials. But இதுவரைக்கும் நான் என்ன புரிஞ்சுக்கிட்டா இந்த மாதிரி ஹோப்லெஸ் பேஷண்ட்ஸ் எல்லாம் டெஸ்ட் பண்ணுவாங்க. பட் ஹெல்தி வாலண்டியர்ஸ் வந்து சப்ஜெக்டா இன்வால்வ் பண்ணுறதுக்கான லாஜிக் வந்து this is the first time I'm hearing that healthy volunteers.
Healthy volunteers.
So far, I have heard health means volunteers.
Human healthy volunteers.
Human healthy
And
volunteers.
when you say human healthy volunteers, they should not be diabetic, they should not be a cardiologist patient, they should be young, they should not be too old. And in some countries, women don't enroll for studies. In countries like Europe and things like that, women come forward to do studies. I worked with a company where we had a plant in Romania, and there the students who are working in the university enrolled for women also enrolled. So basically, you can do studies on men, but is it applicable to women? That's a question. So sometimes, it's not easy to push a lady to come and volunteer for a drug. Men, they do it. But again, men also, you have to actually be honest to tell that in the clinical study, now ஒரு 3 மாசம் நான் ஒன்னும் ஸ்டடி பண்ணலன்னு சொல்லணும். You have to be honest to tell. Because நீங்க ஒரு மாசத்துக்கு முன்னாடி ஒரு ட்ரையல் எடுத்துட்டு இன்னொரு ட்ரையலுக்கு போனா that patient is waste. புரியுதா? அவன் பைசாக்காக பண்றான். You understand? பாவம் எல்லாம் farmers, people who are there, they do it. பைசாக்காக பண்றான். But இதெல்லாம் so இதுக்கு basically there is a called as an ethics group. And இதுக்கு ஒரு database இருக்கு. இப்போ இந்த ஆதார் கார்டு மாதிரி இருக்கு இல்லையா? All the volunteers who are enrolling, then you have to now put your information on the computer, and the computer will tell, based on the information, whether you have done the study before, எப்ப last time study பண்ண, இதெல்லாம் information available in the clinical room. Very important. Otherwise, ரொம்ப கஷ்டம்.
So they look for money.
Basic. That's the fundamental thing. But of course, pharmaceutical companies would be honest to ensure that they're taken care properly. On the protocol here, they are taken care, they're given food properly, they're treated properly. Any other questions? Anything from the technologists?
After now, anybody using ML machine learning in the practice?
Across the board. Today, I can tell you, if you in the US, they say artificial intelligence is reducing the number of manpower in the industry. A lot of people have lost jobs because of artificial intelligence. It's a myth, according to me. Because who is doing the science behind artificial intelligence? Humans, right? So if you don't have humans, how are you going to do it? So it basically, it's a scary situation. A lot of people are getting laid off and things like that. Artificial intelligence is only a tool. Let me be again reiterating, it's only a tool. It cannot replace
Brain.
productivity. Fast.
Fast.
Library is a fast type. Okay, thank you very much. I hope the talk was informative.
Yes, sir.
Maybe it
Informative
is
and
too much
interesting.
I talk. I have the habit of talking too much when I students, but I love my subject, I love my chemistry.
So specifically, it was not superficial. It was very meticulous, and I have got clarity on many aspects.
So you can share your information to your professor. Let me review and come back.
Okay, sir. Okay, sir.
Thank you.
On behalf of the Department of Chemistry and our university, I would like to profusely thank Dr. Ramesh Visvanathan for his informative and interactive lecture. Thank you, sir. I'd like to thank all the faculty members who coordinated for this event and all the students who actively participated in this lecture. I would like to thank once again. Thank you.